Okay, so I'm not sure why organizers decided to ask me to do this talk, and my hypothesis is that being the oldest, I am probably the only one who remembers how to speak without PowerPoint. So I heard it from David Nelson, so I'm not the inventor of it, but that I think is important point about this. So I thought initially to prepare a few slides to demonstrate some things, but then I decided to do it pure. There will be no color images, nothing like this. So the story that attracted me as a physicist to this subject of condensates is that if this is a cell, biological cell, as a physicist thinks about it, it's full of various things here. The most important and the biggest of them is the nucleus, which houses all the DNA, all genetic material. But this is not all. There are, inside the nucleus, there are things called nucleolus, which is, I'm sorry, I don't have time to tell you this story about everybody knows why Newton invented calculus. He needed to derive elliptical and hyperbolic and parabolic orbits. But nobody knows why Leibniz invented calculus at the same time. And in fact, it has to do with this picture in which there is nucleus and then inside nucleolus. So if you are interested, ask me privately. I will tell you the story. It's a wonderful story. So the nucleus size is about 10 micron, just to give you an idea of what is the scale here. It's 10 micrometers. Okay? And there are many other things here. I will not even write down the names of them. Many of them have names. But the point is, when 100 years ago, Oparin and Goldstein and Haldane, speculated about the possible prebiotic evolution, about how life could have started on Earth, they speculated that there should be some protocell, some bubble coacervate, as Oparin called them, that should be surrounded by membranes. So the major idea was that you have to have a compartment separated by a membrane. And then people discovered that indeed nucleus is separated by membrane based on proteins called lamins, so there is some membrane. But nucleolus, for instance, as far as we know, definitely as far as I know, doesn't have any membrane. And many other things in the cell, they seem to be separated, but don't have any membrane. So relatively recently, people realized, I don't know why it took so long to realize, people realized that in fact it could be droplets of a different phase. And there could be phase separation, or better to say phase coexistence between different liquid phases. As I said, why it is so surprising, I don't know, because everyone who ever ate any kind of soup should have known that there is such thing as phase separation between different liquids. Whether it's chicken soup or vegetable soup, there are these little rounded things that float on the surface of soup, and this is exactly the liquid-liquid phase separation. Nothing more and nothing else. So this is biological discovery that there is liquid-liquid phase separation. And some of these things are indeed believed to be different phases, different liquids. This is cytoplasm, and this is some other liquid based on some proteins and so on. So this is interesting now, what kind of condensation this could be. In order to make it a little more specific, let me remind you something that every physicist knows. It would be a phase diagram of a simple liquid, such as maybe water or something else like that. And that phase diagram typically looks more or less like this. So this is critical point. This is gas phase. This is liquid phase. And this is a solid of some sort. Now, important thing that you should remember is that these two lines never end because solid, crystal, and either liquid or gas, they have different symmetries, and therefore the transition from here to here cannot happen without a phase transition, or from here to here. So they never end, while this thing does end at the critical point. And by the way, there are also some lines called spinodals which separate the possibility of overcooled gas or overheated liquid and so on, which is important in the context of this biological context. So I will not be interested in this part, but this part is a prototype of what I will be speaking about. Now, another thing that everybody knows about critical point, that around critical point, there are very strong fluctuations, and therefore the material is very opaque. And I bet that, statistically speaking, looking at the apparent ages of people in the audience, there are people who are painfully familiar with this critical opalescence, because in the lens of your eye, it is supposed to be completely transparent. It is actually the solution of some proteins, many proteins in solution. And as you leave, some of these proteins get damaged. And in fact, the proteins that you have, each of you have today inside the lens of your eye, are the same, not the same type, but the same individual molecules that you were born with. And they get damaged, they get broken. As a result, they approach some sort of a critical point. This solution approaches critical point, and this thing starts scattering light, critical opalescence, and lens has to be taken away in order to give you the opportunity to see. So this is immediately give you some biological application of this. Okay, so another thing that, of course, is known to every physicist is called Gibbs phase rule. And it was discovered by Gibbs in 1875. So what Gibbs said is that in modern language is that the dimension of the boundary in between different phases, the dimension of boundary in phase diagram is 2 plus the number of components in your system, and minus the number of coexisting phases. So of course, in this case, there is only one component, water. So it's 3 minus, let's say, 2 phases, liquid and gas, coexist. And so the dimension of the boundary is 1. This is how it works. But of course, what you realize when you think about the content of the cell is that there are many, many components, many different proteins, and therefore this is not productive. You have huge amount of different components, and it gives no restriction. It's not a productive statement, which is why all the discussions in this condensate meeting are revolving around how to construct the useful phase diagram, because it's multi-dimensional phase diagram, and how to construct it is a big puzzle. So what else do people know in terms of classical physics? What classical physics tells us about this liquid to gas condensation transition? Of course, the classical information about it is associated, I would say, with van der Waals. Again, classical physics, all what I will tell you now, was discovered by van der Waals in 1873, and it was his PhD thesis. It was quite impressive PhD thesis. What he discovered is that, first of all, he realized that this phenomenon of phase transition between liquid and gas can only be explained by interaction between molecules. He was thinking that ideal gas picture of independently flying molecules was clear to him from Maxwell, and then he realized that they should interact. And he discovered something that is now classical, that prototypical interaction between molecules is something like this. There is a repulsion at short distances and interaction at longer distances. He discovered what is called now van der Waals interaction, attraction between fluctuating dipoles, and so on. And as a result, there is this condensation, which he means van der Waals. By the way, I thought all the time that Van was his first name. Yeah. It turned out that the Dutch last name consists of 3 separate words. Okay, so what van der Waals did is he said, he wrote equation of state that, if you remember your textbook, is usually written in this way. And I apologize, I grew up in the tradition in which temperature is measured in energy units, and so typically I don't write Boltzmann constant, and probably in today's talk I will be forgetting about Boltzmann constant. So if you want Boltzmann constant, just insert it in the proper place. So those are material-specific parameters, and this B has units of volume, as you see, and this B has to do with this short scale repulsion, while A is mostly due to attraction. So if you allow me to manipulate this a little bit algebraically, first of all, I will rename this B to be little v because it's just volume of one particle. And then I will say that n, total number of particles divided by total volume of the system and multiplied by volume of one particle, I will call it phi. This is volume fraction of the material. And then my equation of state will be rewritten. No KB here. So that will be phi divided by 1 minus phi minus something that is based basically on this A, and for the reason that you will see a bit later, I will call chi. Okay, so this is a beautiful equation, which, by the way, it doesn't have any derivation per se, because it is just interpolation. It's just a guess. The way you guess it is to say that phi cannot be larger than 1, and so it should blow up when phi approaches 1. Okay. Small phi should be proportional to phi because it's ideal gas. And by the way, this means that there is this power means that this is two-body interaction. Two-body interactions. Okay? So what this means is that pressure as a function of density at small chi, when there is no significant interaction, it goes like this. Then there is critical point, and then it goes like this. And yes, this theory does predict negative pressures when attraction is true. So critical point, you can find that critical point corresponds to this chi parameter to be 3/2 to the power 3, and critical density is 1/3. Finally, you can also easily find free energy per unit volume, also in units of temperature, or KT if you prefer, and this goes the following way. It's okay, so this is free energy of van der Waals gas. Nobody in the field of biological condensates ever uses this equation. Never. The reason is that there is much better interpolation equation that people use, and I will explain to you why they use it. So the much better equation is called Flory-Huggins approximation. And this is something that everybody uses. Flory-Huggins. And by the way, it's interesting that Flory-Huggins discovered this in 1941. Huggins was first. He published his result in May. And Flory was much later, in August. Okay. Sorry?
That's why he's first.
Okay. I don't know why Flory is first. Okay. So what is Flory-Huggins? In the same style, I would write that Flory-Huggins predicts pressure as a function of density in the following form. It's minus log 1 minus phi minus chi phi squared. So Flory-Huggins denoted this energy parameter in units of kT as chi. It's called chi-Flory parameter or Flory-chi parameter, which is why I used chi here. Okay? So it's the same thing. It does blow up at dense packing. Small density, it is still an ideal gas, so it's same thing. But it does not blow up quite that strongly. So if I plot pressure as a function of density, for instance, van der Waals would go like this, and Flory-Huggins would go like this. So Flory-Huggins works better. It works better from the point of view of experiment, and it also works better in terms of basics of statistical physics. If you consider lattice gas, then the pressure would diverge as logarithm, not as power. Okay, so free energy for Flory-Huggins. Free energy is a very nice expression, which is not so different from poor van der Waals. He was almost there, but okay, so attraction part is the same, literally the same. Repulsion part, in fact, the only thing that is absent here is this one. And this allows you to give a very nice interpretation. So what is this? This is, of course, free energy of an ideal gas, and this is actually entropy of translational motion, entropy associated with translations of all the particles. This part, here, it's sort of a guess that it should blow up. But here, you can give it a much better interpretation. You say that this is, if phi is the density of your particles, then you can say 1 minus phi is the density of vacancies. If you think about a lattice model, then this is density of vacancies. So what that is that mixing entropy or translational entropy of vacancies, of empty spaces. And then, if you think about something practical, as Flory-Huggins, in fact, were thinking about polymers, they were thinking not about proteins or anything biological, they were thinking about technological simple polymers that were just discovered not long before their work. And their idea was to say that, okay, and this is quite amazing that they indeed, independently, as far as we know, came with this idea. Their idea was to say that translational entropy of chains is just reduced by the factor of n. If your particles are connected in chains, n particles in one chain, then this is what you have. This is your mixing entropy for the chains. It's just a reduction by the factor of n. But moreover, you can think about mixture of two different polymers, which is what you have frequently in biological applications. Then you have a reduction of entropy of your vacancies that goes like this. And interaction between units is not affected by the presence of bonds, because when you have, let's say, this polymer and another polymer of maybe different type, and what happens is your interactions happen locally in this region, and these shaded monomers and open monomers, they interact in the same way as if they were not polymerized, if they were not.
Derivation, it's from some physics, or it's just phenomenological guess, or what it was?
Well, derivation was, as you can guess, this is some sort of a combinatorics. So there are several pages of combinatorics, but trust me, the price of this combinatorics is exactly as much as my words here. So it's sort of a mean field argument. The deep underlying physics of this mean field argument is something that later Dejean called Flory theorem. There are different things, Flory theory and Flory theorem. So this is Flory theorem. The meaning of this Flory theorem is that when you have condensation of this polymeric material in dilute phase, which is like a gas, your polymer chains fluctuate, and each polymer chain looks pretty much like a random walk, like a Gaussian random walk on average.
What are NA and NB?
Sorry?
What are NA and NB?
So NA is the number of units in this polymer, and NB is the number of units of that polymer. And I imagine that the whole thing is full of these polymeric materials, such that phi is actually phi A, the volume fraction of A material, and phi, 1 minus phi is phi B.
Perhaps I missed it, but what is F? And FB over T?
F is free energy per unit volume. V is volume of one monomer or volume of one unit in lattice model. And T is what many of you would prefer to call KT. Yes.
But this NI and B part are only different by substitution phi to 1 minus phi, right?
To the extent that this is an incompressible object, one of them has density phi and another 1 minus phi. Yes.
Does it implicate the longer is the polymer chains, the smaller the effect of the entropy?
Absolutely. Thank you very much. This was exactly my next statement. Yes. The bigger is polymerization index, the smaller is contribution of entropy. It is indeed true, and this is a big, at the time it was considered a big discovery, but indeed it is physically very obvious. Indeed, if you, for instance, want to know about phase separation between A polymers and B polymers, if they are mixing or not. It's very difficult to understand, but from this formula, it becomes clear that phase separation, as in the case of van der Waals. Remember, case of van der Waals, the phase separation was driven by increased chi, which means by increased A, just renaming of things, and A is this. So phase separation is driven by gain of energy and loss of entropy. So here again, the same story, you gain energy if A, for instance, attracts to A, and by the way, it's useful to say that Flory-Huggins, they wrote this chi parameter in a way which I think will be clear without much explanation. And here, for instance, if I were to write KB, you would ask me, is this B the same as this B?
We wouldn't ask that.
Some of you will. Okay, so this is basically to which extent AB contact is energetically more favorable than homo contact. Okay? So this is energy gain. And entropy loss, if you move, for instance, you separate two chains. Let's forget for a moment the difference between N and A and B. Suppose it's the same N. And then when you separate two chains, you gain energy, some amount per monomer, per 1 unit like this. And you lose entropy about 1 unit, 1 KT of free energy per the whole chain. Oh, this is very natural, and mathematical result of it, mathematical expression of it, is that critical point, this very critical point, like this, where this whole phase separation starts, this critical point with NA and NB, it's simple algebra, how you derive it. Okay, so if both of these ends are 1, then you obtain what? 3. Okay. What did I write here? Oh, I did not write it here. Sorry. It's not 3. It's 2. 1/2 plus 1/2 plus 1. It's 2. My arithmetic is shaky, so it's 2. And critical density is 1/2. And in this case, critical density, I remind to you that this is density of A. So this critical density is actually NB to the 1/2 divided by NA to the 1/2 plus NB to the 1/2. Okay? So, for instance, if you have, if one of the polymers is very long and another is short, for instance, if NB is 1, if NB is 1, then phi critical is, critical density is 1 over square root of n, basically. Okay? Now you may want to realize that this critical density of 1 over square root of n is, in scaling sense, is the same thing as the density inside one polymer coil. If you have a coil which is like a random walk of the size of n monomers, then the size of it will be square root of n, just like the Brownian trajectory of time t is proportional to square root of time, so it's the same thing. And density will be 1 over square root of n. So that's what it means. More importantly, this one means that you need only very small amount of energy in order to condense your polymer molecule. You don't need much. It's only 1 over n, the amount of energy. Again, because when you separate polymers, such as in the case of the cell proteins, you lose entropy only per chain and you gain energy per monomer. So this is the basic physics behind this phase separation. Okay, so what did I forget to say in this? How much do I? Okay, so starting at this point, I will use the rest of the Blackboard to tell you a few examples where some of the more modern, you see that in my description, the latest was Flory-Huggins, 1941. So fast forward to 2000. 2000, and I will tell you, starting from 2000, I will tell you a few ideas from the toolbox of more modern polymer and soft matter physics that are either useful or potentially useful for thinking about these biomolecular condensates, in particular the ones associated with the cell nucleus. So one thing is that, of course, many of the proteins in the cell are charged. And there is also RNAs, which are negatively charged. So the most natural thinking about charges is based on the idea that, okay, condensate should, of course, be neutral. This is very true, or should be overall neutral. However, if you count only the charges on so-called macro ions, which means on ions carrying large charges, such as polymers, charged polymers. If you only count charges on, let's say, proteins and RNA, then is it always true that they should be charged? And the answer is not. The answer is not. They should not be always neutral. And the most sophisticated results on this subject were developed around the year 2000 by my friend and colleague, Boris Shklovsky. Many of you know him. And his ideas I will illustrate by a few examples. So one example would be the most simple and the most sort of for illustration. Suppose that you have a ball of the charge minus Q, negative charge minus Q, and you can of course imagine that another ball of charge plus little q will be attached to it. You can imagine that there will be another charge plus q attached here. Now the question is how big the little q charge can be and still be attached. Okay, so this is a little surprising. To be attached, this q, little q, should be as big, can be as big as 4 of big Q's. everyone who knows the Coulomb law can very easily check, write the Coulomb, not even free energy, but just the energy of this thing, and discover that this is the case. Okay? So this is because, this happens essentially because these two guys are far from each other. So their repulsion is suppressed and they attract better to this one. Another example, more serious example, will be like this. So imagine that you have a negatively charged surface. So I remind you, in the context of condensates, what I'm talking about is how charges can realize attraction. Of course, everyone understands that plus attracts to minus, but how much of pluses can be attracted to a big surface of minuses? This is the question. So suppose that at initial point you have the number of positive charges that are attracted here are exactly compensating. So it is exactly like what your naive thinking about neutrality suggests. Okay, and imagine now that a new plus appears. What happens to him? It turns out that because these things is already sitting there charges, they are strongly correlated. You can estimate that if, for instance, the charges of these ions are, let's say, +3 for each, or +4 for each, then the repulsion energy between these two ions is large. And therefore, they do not want to just smear around. They form what is called Wigner crystal. It doesn't have to be crystal, but they are strongly correlated. And therefore, when the new one appears, it, of course, repels them. And in the end of the day, what happens is that they become just a little bit more closely positioned to one another, and they allow somebody else to come in. Okay? So this is a mechanism of overcharging. Just as in this case, it overcharges. So there is a big sophisticated theory on how much of overcharge you can achieve and so on and so forth. And this is something that you see all the time in these condensates when you have RNAs and proteins, or positive proteins and negative proteins. And this is something that happens all the time. Another way that I like to explain this belongs to Tuan Nguyen, who was actually a student at the time when he invented this way to think about it, is the following. So his thinking was, actually, how much time do I have?
5 minutes.
5 minutes, okay. Then I will not tell you this part. Tuan Nguyen, you can check it. It's published, so I'll tell you something else. Okay, so all of this was equilibrium. Now out of equilibrium. Out of equilibrium. And I'll give you two examples out of equilibrium. One thing out of equilibrium is, imagine that you have some catalytic particle, and you have two types of molecules. somewhere in infinity you have infinite supply of molecules A and B, and A we call fuel, and B we call exhaust or waste. So the idea is that A diffuse here. This is a catalytic particle that transforms A to B, and as a result, there is a flux. And a concentration of A is in steady state like this. If this is chemical reaction rate, okay, if J is chemical reaction rate, you have this type of concentration profile. This concentration profile, in both cases, goes like 1 over R. Now, each of these catalytic particles, in fact, leaves, for instance, if there is also somewhere else another catalytic particle. This catalytic particle lives in the field, concentration field, which is proportional to 1 over R, and that undergoes the so-called diffusiophoresis. It moves with the speed proportional to gradient of concentration. Can be positive or negative, but this motion with this speed indicates that there is interaction between these catalytic particles, which is like Coulomb interaction. So based on that, you can realize the whole zoo of, and this is mostly Ramin Goleshtanian. So you have the whole zoo of non-equilibrium quasi-Coulomb particles, systems. And now, so this is example 1. Example 2. Suppose that instead of just one temperature, suppose you have 2 different thermostats. 2 different thermostats. This is a little bit artificial on the surface of things. However, for the biological context, you can imagine that some of the particles are driven by some activity. For instance, I told you that the nucleus is full of DNA, and some of these DNAs are presently read in order to synthesize RNA, to read genes and so on. So there is some activity. So they move potentially faster than the surrounding parts of DNA, and you can, to some approximation, you can think about it as 2 different parts of DNA are driven by 2 different thermostats. So what will happen in that case? So I will give you a little bit of algebra. So imagine that you have 1 particle A and 1 particle B, for instance. And this one is driven by temperature TA, and this one by temperature TB. And therefore, imagine that, for instance, they have the same friction coefficient in the water for simplicity. So you have a Langevin equation for particle A. If they interact with some sort of potential U, then there is force minus gradient of potential, and then there is Langevin random force, which is proportional to the square root of temperature, and there is random delta correlated Langevin force. Similarly, there is second Langevin equation. This is now not minus but plus gradient U because it's opposite force. They interact, but it's the same force acting in different direction. So square root of 2 zeta TB. And there is another statistically dependent Langevin force. Okay, now subtract one from the other. So you see that the distance between them satisfies now minus 2 grad U. Okay, and now when you add together Gaussian variables, then what happens is you obtain squares of the amplitude and sum them together. So you obtain the root of 2 zeta Ta plus Tb. This is the biggest algebra that I will use. Okay? And this is what? This is Langevin equation for a single particle in the potential 2U and at temperature Ta plus Tb. So this converges to Boltzmann distribution of the distance between two particles, and I will not write the normalization factor. So this is U divided by twice, by half sum of Ta plus Tb. So this is like a Boltzmann distribution, but with the new temperature. If these two friction coefficients were to be different, then algebra would be slightly more challenging, but still doable, and the result will be like this. Okay, so going further, you discover that indeed this type of A and B particles phase separate under certain circumstances. They phase separate if the ratio between Ta and Tb is sufficiently large. Namely, White in Waals theory showed that the ratio should be bigger than this strange number. I once...
Why it's known that if it's a non-equilibrium, why the temperature does exist?
Well, it's a model. It may not, the model is not valid for anything, for everything, but in this model there are two temperatures. And
yeah, but I mean, when it's equilibrium, then, would be...
There would be no temperature in general, but there, you see from this algebra that there is this Boltzmann distribution with this. It's not temperature, by the way. What is this? This is...
I see.
But I spoke once about this, and there was a mathematician in the audience who said, this is like this. Okay? All right, so this is huge number, so this is unrealistic. But remember, when you combine units into a polymer, things become much easier. And it turns out that if you take if you polymerize your particles together, then it is something much more modest. And this way, you can probably do it much cheaper. You don't have to have so much of temperature contrast. Why it is 1 over square root of n here and 1 over n here is something that we're trying to understand, because this is computer simulation by Kurt Kremer and Jan Smrek. And there is no understanding of it. So, okay. Finally, as I told you, in the nucleus there is this nucleoid, and there is different types of chromatin. That chromatin that is presently red, which is called euchromatin, and heterochromatin. So, is it possible to interpret the nucleolus and separation between chromatin, euchromatin, and heterochromatin using this? Is an open question. So I stop with open question, and I apologize, Lars had to stand for so long.
Please. Just a question there. Do they space separate because there's a high difference in motility? The one that's moving just a little bit, the one that's moving a little bit?
They separate because they move with very different ways. One way to think about it is to say that, imagine that you have this system of very hot particles and very cold particles. So cold particles will be outside, and you form a bubble with very hot particles. So the idea would be to say that in order for pressures to be the same, cold particles have to have much higher density than hot particles. Hot particles will realize much bigger pressure at low density, and that's how phase separation works in qualitative terms. But in general, of course, in order to see this, you have to solve the kinetic equation and so on, because you don't have any thermodynamic tools as soon as you have non-equilibrium systems.
Please.
Do you think that TA and TB is maybe a little hard to think as realistic? Think that you could replace it with masses and get a similar, like one more massive than the other one, and because the TA and TB are kind of playing a role of like a diffusion constant, so you could just say, well, how else can I change the diffusion constant? One would be by changing the masses.
Well, as you know, masses do not enter into equilibrium statistical mechanics. So masses, no. There should be some activity involved, because you can realize that their detailed balance in this system is broken. Although this very much looks like Boltzmann distribution, but in fact, more close analysis of it shows that the way A approaches B and then the way A goes away, they are different. So detailed balance is violated.
Please.
Yeah, hello from the other program. So you keep talking about separation of the phases. So does that mean that we are born as a happy emotion and then at some point it separates, or like why?
Well, I don't remember how I was born. Probably, I would guess I was not born as a uniform solution.
Emotion, but I said, yeah.
Well, I started even one step further.
So why, I'm sorry, then let me rephrase. So why,
No.
why is it a big deal that phases are separating then?
I cannot agree more with you. I agree it's not a big deal, and the big deal is not the fact that there is phase separation. The big deals are in details. what kind of, for this type of proteins, they separate and they do this and that. Or nucleolus. Nucleolus, the ribosomes are made inside nucleolus and how they are made, and this is all big deal. Or separation between heterochromatin and euchromatin. Both are chromatins, so any type of consideration like Flory-Huggins and so on doesn't seem to apply, so there is probably non-equilibrium, maybe this type or maybe some other type, some non-equilibrium separation. Those are maybe potentially big deals, and people who work on that are convinced that they are big deals. But I totally agree with you that as a matter of principle, the fact that there are phase separations, as my example of soup suggests, it's not a big deal.
So sure, let me ask you a different question. Often in these fields, some of the functions learned by disease, so are there known chemical reactions or diseases that lead to the loss of something as a functional loss, and is that often how these things are become more relevant than to the physicists? Maybe you're not the one to answer.
I'm not the one to answer. I know the answer is. I know the answer is yes, but I would not prefer to expose my ignorance any further. So please, anyone.
Maybe I can add a comment. So going back to your question, so in addition to sort of like thinking about like these compartments inside cells forming via phase separation and forming these biomolecular condensates, as Shura was mentioning, there are important consequences for cellular function, but there are also consequences for cellular dysfunction. So what has been shown in some examples is that over time, these liquid-like droplets inside cells, they can change their properties, and they can become sort of like solid-like inclusions that are implicated in things like neurodegenerative diseases, like ALS, frontotemporal dementia. So like not only what drives proteins and nucleic acids to assemble into these structures within cells, but how their material properties are encoded and how these properties can change over time.
You can remember also the example of cataract.
Yes.
Steph, did you want to add?
Yeah, and I mean, the big deal, I think, is that all biology textbooks say that you have compartments that are separated by membranes. And that is a really energetically consuming process to create membranes, and it's slow for them to be dynamic. And so the fact that you can have this obvious physics in a cell, and that it can maybe do something to the cell, and the cell can harness that, that's the big deal. That's the exciting thing. That you can get, it's another mechanism for compartmentalization of the cell.
Great. Okay, thank you very much.